Okay, welcome to chapter 24-4, the stomach. This is one of my favorite organs to talk about because it has so many different functions and a very coordinated system of events. And it's a really cool organ because we manipulate it a lot. We use it on the daily basis. There's just a lot going on to the stomach. But before we jump into it, I want to remind you of a couple of things. First, we're going to be talking about hormones and enzymes. And I want you to remember that hormones can either be peptide hormones or steroid hormones. But either way, these are typically going into the bloodstream and finding their target organs or tissues and then activating some sort of response. Enzymes might not necessarily be their activated form. Sometimes they have to be converted into their activated form. So those that end in ogen are inactive. And then those that end that don't have ogen, they just don't have ogen at the end, or maybe they have a different prefix. Those are usually the active forms. Okay, and we do this because we don't want the inactive forms, we don't want the active forms coming out of their respective tissues, because if they were active, they might actually deteriorate the tissues that they're coming out from. So we'll try to make them inactive as they come out of the tissues that they're not affecting, and then turning them into the active forms whenever they finally reach whatever environment they're supposed to reach. The second thing I want to remind us all is of our equation we learned from the respiratory system chapter. So after cellular respiration for tissues, they will really make a byproduct of CO2 and water. And then with the help of carbonic anhydrase, we can have a reversible reaction with an intermediate, H2CO3. And that intermediate can also change with the help of carbonic anhydrase into the products H plus protons and HCO3 anions. And remember, this is a really important proton because that can help us determine acidity of an environment. And we talked a lot about how CO2 is involved with the respiratory system. But really what I want you to focus on is the hydrogen proton, and don't forget that this equation exists. Let's see. The other item that I want you to recall, that's it. All right. So now let's jump into it. So our stomach. Our stomach is a J-shaped organ, and it is coming just off of the inferior aspect of the esophagus, so J-shaped like this, woohoo, and a little ending piece, and then dumps into the small intestine. So we have our esophagus, and then our stomach, and then the stomach dumps into the small intestine. And the small intestine comes in 3 parts, which we'll talk about in the next section, but the first part being the duodenum, so you'll hear me say duodenum a couple of times. The functions of the stomach, we're temporarily storing food. Once we're storing the food, then we're going to start mechanically digesting it, so the stomach is going to do some muscular contractions itself to actually mechanically break apart the food. And then we're also going to chemically digest the food with the help of some enzymes. The end result is chyme. So we started with bolus up in the esophagus, and then in the stomach we start creating chyme, and by the time we're done with digestion in the stomach, then we end up at the end of the stomach, and that chyme then transitions into the small intestine. Remember, we have the lesser curvature of the stomach and we have the greater curvature of the stomach, and we talked about how the lesser curvature has the lesser omentum and the greater curvature has the greater omentum, and those will be those are important topics that we've already covered. Next, we need to talk about the regions of the stomach. There are 4 of them. The first is the cardiac region, or the cardia region. This is a difficult space to actually really identify with just one pin, but it's typically where the bolus will first hit the stomach. Then we have the fundus, which is the upper top portion of the stomach. Then we have the body of the stomach, which is the main portion of the stomach. And then we have the pyloric part of the stomach, or the pylorus. There are lots of structures within the pyloric part. Don't let any of the names confuse you. You already know anatomically what these structures mean. Antrum is an entry. Canal is like a duct. Pylorus is just the area that we're talking about. And a sphincter is a type of constrictor muscle or a circular muscle. So but just add pyloric or a pylorus at the beginning or ending of each of those to describe those anatomical structures at that part. We also have a lot of rugae within the within the inside of the stomach. These look like wave-like structures, and the cartoonistic depiction is actually really similar to what it looks like in real life. And the whole purpose of rugae is to increase the amount of surface area we have, A, and B, to allow for to accommodate the amount of stretch we might have if we eat a really, really big meal. Something that's also special about the stomach is we have the 4 layers of the digestive tract here, but our muscularis layer actually has a third additional muscle that's deep to the other circular and longitudinal muscles, and that's our oblique muscle. And essentially, why the oblique muscle is really important is because while the circular and the longitudinal muscles are working in a perpendicular plane to each other, the oblique muscle also comes in and allows for the most amount of mechanical digestion as possible and breaking down of the food as possible. And what ends up happening is the stomach actually kind of wrings out itself, similar to like a dish towel that you'd be wringing out of water. I want to pull up Complete Anatomy here so that you can get a nice view of what's going on with the stomach. So here we have all of the digestive system highlighted. I'm going to go ahead and remove some structures so that we can, or I'm going to remove the greater omentum so that we can get a better view of the stomach. That's right over here on the left-hand side. I'm going to go ahead and remove some organs so that we can see the stomach clearly. So we can see this darker maroonish color organ. That's going to be the part of our large intestine. And then we have this pinker set of intestines. Those are going to be our small intestines. And what's left is we have our liver over here on the right-hand side. I'm going to go ahead and remove the skeletal system so that we can get just a nice clear view of the stomach. I'm going to remove some ligaments and then our ciliary tree, which we'll talk about with the accessory organ section of this chapter. Okay, so now we're looking at the anterior aspect of the stomach. Remember, the esophagus is where we're coming off of. So the cardiac portion is not labeled in this app, but it would generally be in this vicinity. Then we have the fundus of the stomach. Then we have the body of the stomach. And then we have the pyloric part of the stomach. I'm going to flip us around to the posterior aspect and identify one other organ, and that's going to be our pancreas here. The pancreas is really important in digestion, and we'll talk about why. But for right now, I want you to appreciate that the pylorus is kind of hugging the posterior aspect of the stomach and is immediately touching the beginnings of the small intestine, which is the duodenum right over here. So I'm going to flip us back around. And now I'd like us to open up the body of the stomach. I'm going to hide that and reveal some of the layers of the stomach. So just like in that cartoonistic depiction, we have the longitudinal layer muscle, we have the circular muscle, and then deep to that we have the oblique muscle. And so now you can appreciate if we were to contract any of these, like if we were to contract this muscle in that direction, contract this muscle in that direction, and contract the obliques in this direction, we would have more of a wringing out of the entire stomach. And then within it, we have our rugae, our folds, and at the end, we have our pyloric sphincter. And I'm going to kind of draw that in here, and you can imagine if it was closed all the way like this, that would be our, oh goodness, oh good, okay. That would be where we have that last, or the end of the stomach will have that sphincter. Don't forget, at the top of the sphincter, we also identified the esophageal sphincter. This is not a true sphincter. It's just shaped like a sphincter, and it's relatively pretty good at keeping the acid of the stomach in the stomach instead of letting it come back up as gastric reflux. And so here I want to identify that pyloric sphincter. And this is really important because once the chyme is churned in the stomach, we want to keep it in the stomach for a period of time. So this pyloric sphincter is going to close itself off in order to allow the stomach to hold on to its contents for a period of time before we allow it to dump out and down into the duodenum. Okay. All right, let's go back to our lecture slides. All right, so what kind of tissues do we have in the stomach? Simple columnar epithelium. And sandwiched in between each of these columnar epithelium, we're going to have a lot of mucus cells. And these mucus cells are really important, or goblet cells in order to produce mucus. I'm sorry, not goblet cells, just mucus cells to produce mucus. And that's really important because we have a really acidic environment that we're about to deal with. So we need to prep the stomach with a basic environment so that we don't ruin the actual lining of the musculature. Then we'll have these things called gastric pits. The gastric pits are pits within the rugae that are also secreting mucus, and deep within the pit we'll have gastric glands, and those gastric glands are going to be secreting gastric juice. Gastric juice is going to be a combination of enzymes and compounds that are going to be really important for us to help start the breakdown of proteins. And so with that, let's talk about those three important cells. So first is the parietal cell. Parietal cell. And the parietal cells are going to be secreting two things. The first thing is hydrochloric acid, or HCl. And hydrochloric acid is going to be really important because that hydrogen proton is going to allow for that really acidic environment, which will be important here in a second. The second thing that it's secreting are intrinsic factor. Intrinsic factor. And intrinsic factor is super important because intrinsic factor is the only way humans are able to actually absorb vitamin B12. So what does that mean? Whenever we're digesting food and we have any vitamin B12 in the stomach, that vitamin B12 can't diffuse past the absorptive epithelial lining in the digestive tract. It has to be carried with something, and that something is intrinsic factor. So intrinsic factor, whenever it sees vitamin B12, it'll hang on to it like best friends, and it'll help carry it through the absorptive lining so that we can actually do something with it. I'll abbreviate parietal cells as P, but this is not an appropriate way to abbreviate parietal cells. It's just a space thing for me so that we can get a lot of stuff on one picture. So please don't shorten parietal cells to P cells. That is not appropriate. The next cells we need to talk about are chief cells. And same thing, I'm going to call these C cells, or I'll draw these in as C cells, but again, please don't identify them as C cells. Chief cells are going to be secreting pepsinogen. And pepsinogen is a really important non-functional enzyme, and we know that it's non-functional because it ends in ogen, so the actual active form of it is going to be pepsin. And as you can imagine, it has pep in the name, so it's probably going to be breaking down some peptides, which are proteins, and we'll talk about how that happens shortly. And last but not least, then we have some G cells, and these cells are the only ones, or so far, that you can call actually G cells. I forget what the G cell stands for. It might be gastric cells. I don't necessarily remember, but you can call these G cells, and the G cells are going to be secreting gastrin. Gastrin is actually a hormone, and remember, with hormones, those are going to be traveling in the bloodstream. Okay. So to hold on to this, we'll bring this back up here again shortly, but let's take a look at what those gastric glands actually look like. So here we have a cartoonistic picture in the middle of the stomach wall. We have the rugae. In the rugae, if we looked deep, we could see the gastric pits. If we looked even deeper, then we could see the gastric glands. And you can see in the gastric gland picture, we have the parietal cells in kind of an orange, the G cells in a yellow, and the chief cells in kind of a salmon colored. All three of these cells are going to be secreting gastric juice at the same time. That's going to get pumped into the gastric pit, and then it'll eventually make a kind of a lining across the rugae, kind of like a gelatinous layer almost, but less gelatiny. And on the left-hand side, we can see a microscopic picture of what this would look like. So here we don't get quite a nice view of the layers of the muscle, but focus on the gastric glands. This is a relatively thick layer. We've got a lot of cell activity going on. And then in the gastric pits, we have a lot of structural support here with those columnar epithelial cells. Okay, so let's talk about parietal cells really quick. Parietal cells are going to be secreting one of two things, right? They're going to be secreting HCl or intrinsic factor. Let's talk about how it secretes HCl, because how are we secreting an acid whenever it's an acid, and acids tend to destroy tissues, right? How do we actually get that acid out into the stomach? So in this picture, we have a box of a parietal cell, and on this side we have the lumen, and on the other side we have like what would be the bloodstream. And we need to start with something that we're familiar with, which is going to be carbon dioxide and water. And remember, these are going to be the byproducts of cellular respiration. So these are happening in the cells all of the time. And carbonic anhydrase is also floating around all the time. So, and we don't like storing carbon dioxide like this. We like to store it in a less charged form. So we're going to store it as H2CO3. But H2CO3 can also dissociate, as we know, onto the right side of the equation into HCO3 and hydrogen protons. Okay, so that's all fine and dandy, but we don't want an accumulation of hydrogen protons living in the cell. So what are we going to do with those? We're going to pump those out into the lumen so we can get rid of those. The HCO3 molecule also has to go, or the compound also has to go somewhere, and that tends to go back into the bloodstream. And that'll be really important because it'll go back into the bloodstream to help pick up additional hydrogen protons to help buffer whatever environment the bloodstream might actually be carrying. But HCO3 can't just dump itself into the bloodstream on its own. It has to actually be co-transported with something or swapped with something else, and that something else is chloride. So we have this fancy little protein channel that will accept HCO3 going in the left direction, going back into the bloodstream, only if we can exchange it with chloride. So chloride ions are also floating around in the blood at any given time, and we're going to pull those into the parietal cell with this exchanger. Where do these chloride ions come from? That's a great question. Stay tuned for the urinary system. We'll talk about that there. So anyway, we have some chloride ions in the parietal cell. That doesn't really seem that great because it's just floating around on its own. It's a relatively charged ion. That doesn't really seem like such a great choice. So it's going to look for its hydrogen. Oh goodness. It's going to look for its hydrogen ion, proton, on the luminal side of the parietal cell, which happens to be the stomach. So we have our hydrogen protons, we have our chloride anions, and together they're going to make hydrochloric acid in the lumen. So that's what this picture is depicting. I have two different arrows trying to separate the two different stories that I just talked about. If the 1, 2, 3, 4 in the book makes more sense to you, go for it. But it's just important to understand where we're actually getting these relatively charged and really acidic acids from. So that's how we get hydrochloric acid. Okay, you're talking a lot about acid, you're talking a lot about hormones. I don't really get why we're supposed to care or how these all come together as one story. So that's what this picture does for you. This is from your Spotlight figure. It's number 24-15 as one of the images in your book. And it goes through the 3 phases of digestion. And this is a really, really, really important story, if not one of the main stories that we'll talk about with this chapter. So you really need to become familiar with it. I'm going to go through one by one with the cephalic phase, gastric phase, and intestinal phase here in the next couple slides. But this is the picture that you want to go back to and reconfirm that you really know what's going on when you're studying this. So let's begin by identifying the names of all 3 of these phases. So 1 is cephalic phase, 2 is the gastric phase, and 3 is the intestinal phase. And the naming of these makes sense, right? Cephalic, we're going to start somewhere with the head. Okay, that's cool. Gastric, then we're going to go somewhere in the stomach. Okay. And then intestinal. Okay, so then we're going in the intestines so that we can actually start absorbing things. Cool. Okay. So cephalic, all right, like that's the beginning of digestion. That doesn't really make sense. I thought whenever food actually gets to my stomach, it's the beginning of digestion. Well, remember, some things are actually starting the cephalic phase that we might not even be like consciously aware of. So once we start thinking about food, smelling food, hearing food sizzle on the grill, we actually start salivating with those salivary glands in order to prepare the mouth with that mucus and water in order to accept whatever it is that's coming from the outside environment in. And we're also going to produce some mucus out in the stomach as well so that we can protect the lining of the stomach before all of that food stuff comes in. What we wouldn't necessarily think about that's happening in the cephalic phase is what's actually happening in the stomach. So in addition to the mucus, we also want to, let's go in this color. So in addition to the mucus, we also want to start prepping the stomach with some of the enzymes that are already being released so that by the time the food actually arrives there, we can be really efficient and start breaking it down immediately. So I'm going to put mucus and enzymes down here. Okay, so now let's draw what's going on in, let's draw what's going on in that picture here down below. So I'm going to draw a stomach. a terrible picture of a stomach, but a stomach. And I'm going to zoom in here at the top part, and I'm going to abbreviate P for parietal cell, and I'm going to abbreviate C for chief cell, and the other cell that we learned is the G cell. I'm going to put that down here. Now, remember that we have that those gastric glands, and they're going to be everywhere in the stomach. The reason why I'm drawing it like this is because I think this is a little bit more like chronological, and it makes sense to me. Like food is going to come in from the esophagus and eventually go down into the stomach. And the parietal and the chief cells are probably going to be activated a little bit sooner than the G cells, but relatively all three at the same time because they're all part of the gastric glands. I just like drawing it like this because once we finally finish the entire picture, it'll make a little bit more sense organizing wise why I drew it this way. But just know that these cells are scattered throughout the entire stomach. They're not just located in where I drew them. Okay, so we have the gastric glands. The gastric glands are going to be bumping up right against the submucosal plexus. And the submucosal plexus is being talked to by the central nervous system via parasympathetics from cranial nerve 10. So the submucosal plexus tells all three of these cells to start secreting stuff at the same time. So let's start with gastrin. The G cells. G cells are going to be releasing gastrin. Gastrin, I told you, was a hormone. So it's not going to go into the lumen of the cell, it's rather going to go into the bloodstream. So I'm going to draw it coming outside of the stomach and into the bloodstream. I guess I could draw some bloodstream out here. Okay, and then our parietal cells are going to be releasing two things, one of which is intrinsic factor, and the other which is HCl, or hydrochloric acid. Okay, and then the chief cells are going to be secreting pepsinogen. And like I said, pepsinogen is not the active form. We have to turn that into pepsin, but for right now we're going to be secreting pepsinogen. We'll talk about how pepsinogen actually becomes the active form here in the gastric phase. So this is going to be, and of course we got some mucus floating around down here as well. Got some mucus also in the stomach. Mucus. So now we've prepped the stomach. We're thinking about food. We put food in our mouth. We ate it. We chewed it. It's going down into the stomach. Good news. Let's move into the gastric phase. So this is going to take a little bit more time because now we actually have to start breaking apart those large portions of the bolus whenever it comes down the esophagus. So the gastric phase gets initiated by three things. One, the actual distension of the stomach. So the actual spreading out of those rugae, the spreading out of those folds will actually help initiate more contractions of the stomach and helping move food down the esophagus and into the stomach. Alkaline contents. So if we have a bunch of mucus in there, we're going to have a way more basic environment, and that's also going to trigger the stomach to say, oh, okay, I should probably get some acid in there too so that I can help break apart some stuff whenever we finally see it. And then 3, the presence of peptides and proteins. So remember, we have the 4 macromolecules of life, and we'll talk about this many times in this chapter. We have the 4 macromolecules of life. And that's going to be proteins, nucleic acids, carbs, and lipids. Okay, and we have polymers of these and we have monomers of these, but let's for right now focus on the proteins because, like I said, the stomach is focused on breaking apart proteins. And so proteins, their polymers are going to be peptides or polypeptides versus their monomers, or the smaller version of them, are going to be the amino acids. And so our cells, whenever we actually get to absorb these molecules, we can absorb really, really, really small peptides, but we really like absorbing amino acids. That's much easier for us to actually work with rather than peptides. So we're focused on trying to get our big proteins, our big steak that we just ate, down to amino acids. Okay. Stimulation. So I have these numbered for each of so stimulation. 1, histamine released by ECL cells promotes parietal cells to release hydrochloric acid. So the trigger was the distension of the stomach, and the stimulation from that trigger is going to be histamine release. Same thing goes for number 2, and then the same thing goes for number 3. So for both 2 and 1, neural short reflexes to initiate mixing waves by the muscles is going to be the response for the stomach sensing that there's alkaline contents. Number 3, the G cells start releasing more and more gastrin whenever they start to see more and more proteins and peptides. And then the final result of all 3 of these things happening is we finally get more hydrochloric acid, we get more mixing, and we get more gastrin, and more mucus and pepsinogen production. So the entire idea of the gastric phase is not only did the cells already start secreting what they were supposed to secrete, but now they're in hyperdrive and they're going to keep secreting everything that they need to secrete with the goal to break down everything so that by the time the chyme leaves the stomach, it's been broken down as much as possible. That's the whole goal. So here's the picture from your book. So now let's add a little bit to our, just make sure. Yep, okay. So let me add to our chemical picture over here on the right. So I told you that pepsinogen is not the active form and that the active form is pepsin. All right, so how do we actually get to pepsin? So we have to wait for the environment of the stomach to actually reach about a pH of about 2, which is a really, really acidic environment. And so how are we going to do that? That pH is actually going to come from the continuous dumping of hydrochloric acid. And when we finally get enough hydrogen protons, we're going to finally decrease that pH value down to 2. And once we finally get that, then we finally actually convert pepsinogen into pepsin, and pepsin is going to be that activated form that can start breaking down those polypeptides into peptides and some amino acids. So that's happening. Number 2, we have more intrinsic factor being secreted. We have even more pepsinogen being secreted, so more of this cyclic thing is happening. Then we got more hydrochloric acid being secreted. We've got more mucus being secreted. Everything is continuously being more secreted. And of course, gastrin is also continuously being more secreted. The stomach is also being stretched or distended. So we've got stretch receptors saying, oh my gosh, we have more stuff coming in. We need to go, go, go. So it's going to start contracting the stomach even more. Now we also have a more pH or a more acidic environment. And so now the chemoreceptors are like, oh my gosh, it's really acidic. We got to get more stuff in there. So maybe we'll secrete more mucus to balance it out. So this goes on and on and on, almost like a positive feedback mechanism until we finally get all of that chyme in the stomach broken down as much as we possibly can. Okay, so speaking of feedback loops, some of these cells are actually going to have positive effects on other cells. So G cells, and let me draw this in another color. Let me go gray. So G cells, once gastrin is released, gastrin is going to go in the bloodstream. And gastrin is actually going to go and have a positive feedback on the chief cells, and it's also going to have a positive feedback on parietal cells. So that's how we get this continuous pumping of gastric juice by both the parietal and the chief cells. What else do we got going on? I don't think anything else that I haven't already mentioned to you. Okay, nothing else that I haven't already mentioned to you, except now we have to introduce another cell. So I'm going to draw that in blue, and I'm going to put it down here by the G cell. And these are going to be D cells. And D cells are going to release something called somatostatin, or SST. I'll spell that down here. And somatostatin, also a hormone, also going to travel in the blood. But somatostatin is actually how we're going to turn this whole thing off. So somatostatin is going to have a negative feedback or negative effect on the G cells. And whenever it does that, that actually helps stop the entire loop altogether. Because gastrin, as you can see, won't be able to continue its positive feedback loop on both the parietal and the chief cells. So all of that gets shut off. Now we're just focusing on the D cells, turning off the G cells, so we can finally end not only the end of the cephalic phase, but also end the gastric phase as well. And maybe we can finally move on to the intestinal phase. All right, so intestinal phase, we're moving into the intestines. The trigger of this is distension, decreases as chyme leaves. So as chyme finally leaves the stomach, It is headed down to the duodenum, or the beginnings of the small intestine. And as it's leaving the actual space of the stomach and the stomach goes back to its original shape, that will initiate the intestinal phase. And what happens is we begin the enterogastric reflex, which will inhibit the myenteric plexus, and myenteric plexus is what's causing the muscular contractions in the stomach. And then we're going to start turning on some other hormones like CCK, GIP, and secretin, and those cells are going to go and turn off the parietal and chief cells. Even one more further step to make sure that we we induce negative feedback back on those cells so that we can stop secretions in the stomach. We don't need it anymore. We'll talk more about CCK, GIP, and secretin here in a second. And then the end result is the enterogastric reflex stops gastrin, and we finally have contraction of the pyloric sphincter. So now nothing is left in the stomach and everything is getting pushed down into the stomach, or everything has been pushed down into the stomach. And that's our intestinal phase. This is going to take even longer than what was going on in the gastric phase, because in the intestines is where we're actually starting to absorb things. Or absorb our monomers, I shouldn't say things. Okay, so I'm going to go ahead and erase somatostatin, and I'm also going to erase absorbed monomers, so make sure you have that somewhere in your notes. I'm going to continue the small intestine down like this, and now I'm going to add in these 3 hormones. Oh, I don't want to do it in the same color. Let's go in this color. Okay, so we're going to have CCK coming from I cells, and we're going to have GIP coming from, K cells? I can't remember. You don't need to know I or K cells, so I'll just put a question mark here. And then we have secretin, and secretin is coming from a lot of different organs at once. And these are all going to target the duodenum to do certain things, but let's talk about what they collectively do to the chief and the parietal cells. So I'm going to draw an arrow down here. They're going to inhibit the chief and the parietal cells. They're also going to inhibit the G cells, and that will essentially turn off the gastric phase. It will also inhibit the myenteric plexus, so we stop muscular contractions from the stomach, and we eventually get to cinch off the pyloric sphincter or close it off so that we can get all of the chyme into the small intestine. CCK is going to have additional features that we'll talk about whenever we talk about with the gallbladder, and secretin, secretin and GIP are also going to have additional features as well, but we'll get to those whenever we talk about the duodenum. Okay, so we talked chemically how we're mixing and churning things. Let's talk a little bit about reflexively what's going on and neurally what's going on with our we only start with the 2 intestinal phases first, and then we'll talk about the 3rd phase. So, first phase is the gastroenteric reflex. Excuse me, not phase. So gastro meaning stomach, enteric meaning intestines, and it's a reflex. So this is what's stimulated whenever our stomach gets stretched or gets distended. Then once the gastroenteric reflex gets stimulated, we eventually move into the gastroileal reflex. So what ends the gastroenteric reflex? Oh goodness. What ends the gastroenteric reflex is the closing of the pyloric sphincter. And now we can enter into the second reflex, which is the gastroileal gastroileal reflex. So gastro meaning stomach, ileal meaning the ileum, and the ileum is the last portion of the small intestine that is attached to the first portion of the large intestine. And there's another sphincter in between those two, and so we're going to go ahead and open that sphincter, the ileocecal valve, so that we can have stuff from the small intestine actually move into the large intestine. And this reflex ends once the ileocecal valve closes. Okay, so I have a set of questions down here. What initiates each reflex, which I've listed? What order are these reflexes activated in? I've got you gastroenteric and gastroiliac reflexes. And then what phases are involved in each reflex? And so by that, I mean, think about where the cephalic phase would be as part of the first and the second reflex. Where would the gastric phase start and stop between the first and the second reflex? What about the third phase, the intestinal phase? Is that part of, or does the gastroenteric reflex help stop that? Is it happening at the same time as the intestinal phase? What about the gastroileal reflex? So, oh, nope, I don't want to go to Living Social. Okay, so here, compare reflexes to the phases, the three phases. And I won't talk about this over in lecture, but if you have any questions or this doesn't make any sense, please bring it up during tutoring or discussion hours, and we can definitely chat about it.